informative presentation on AIDS and Ebola, as well as the horrors of vaccinations and genetic engineering. I'm Wes Mann of the Preferred Network, and I'm very proud to be sponsoring this event this evening. I'm sure that you would all agree that AIDS and vaccinations are among the most important topics of today. And our speaker tonight is well qualified to speak on these matters, which not only greatly affect us, but will also greatly affect our children. Here to give us the shocking truth of the true origins of AIDS, against all popular belief, medical or otherwise, is Dr. Len Horowitz, a Harvard graduate, health educator, author of 10 books, and in my opinion, a true hero. I know that you're anxious to hear what he has to say, so please give a warm Kelowna welcome to Dr. Len Horowitz.
Thank you very much. It's a privilege and a pleasure for me to be here. I'm not going to be up here for too long up at this podium because I like to have eye contact and connect with you folks. It's not such a huge audience that we have to be separated by this barrier. So I typically don't use this thing. So let's see if this works without me being there. Can everybody hear me okay? Good. What freedom. It's a privilege and a pleasure for me to be here. Thank you so much for taking your time out of your busy lives and spending this next few hours with me and sharing this information and sharing this tragedy, the horror reality. Typically I ask you, how are you doing tonight? And everybody says, oh, we're doing great. And I typically say, well, you won't be doing so well when I get done with you. But I really want you to know that you should hold this information in a greater context of hope, of actual celebration, because the information that I'm about to share with you really represents some of the most horrifying facts, documented facts, that literally represent some of the most painful reality and example of man's inhumanity towards man that has ever been brought out. And so that represents an extremely painful and difficult reality to consider. It's shocking. And that if you don't live in denial about it, like most people are, and if you open up to it, then the pain of that lesson is an extremely strong motive for creating change. Because if you look in your own lives, typically, you never changed unless you were forced through some major pain or slap to change. And that's literally the way God planned it. And so that the opportunity, given the fact that this is the most painful reality that we could ever come to terms with, it should also be kept in the context of celebration, because I was used as an instrument to bring exactly the lesson that God wanted to have out now, so that we can maybe wake up, realize the pain, and move on to exactly where we're in the process, our destiny of bringing in the paradigm shift, the new age, the messianic era. It's really all the same, you see, when we can literally live in love and not fear. And it has traditionally been fear and separation, dividing and conquering the sheeple to conquer the flock. And so that's basically what the context, this is a context of celebration, but yet you who came here tonight, you will be enrolled, whether you like it or not, whether you expect it to be or not, whether you're highly critical, and I urge you to be critical of this information I'm about to give you. Use a lot of discretion, because this is a horrifying reality that you're going to have to share with a lot of family and friends, people you love and who you care for, who you want to save their lives, and you want to bring them into the next century. Then you're going to have to be just like me. See, you think I'm a hero. Well, it's your turn too. And that's why you're here tonight. You didn't know that. But that's why you're here. So welcome. This is a celebration. And we're all in this together. This is now, at the end of the evening, you'll certainly be understanding that this is our crusade together. And together, those who have eyes to see and ears to hear what we have to share here tonight will be the ones who survive and make it into Yisroel, when the lion lies down with the lamb, and when we have truly peace on earth, And we do not have to worry about a military, medical, industrial complex that will manipulate us in every which way possible and literally kill us without us even knowing what they're doing. That is a wonderful opportunity. So, indeed, this is a celebration, but it's a serious celebration this evening. And I want you to know, it will take you, literally, some time to integrate the information that I'm going to present tonight, and that's fine, because, you know, Elizabeth Kubler-Ross's death and dying model, where you have to be in denial first when faced with a horrifying reality, such as a death. It's kind of like a death for many of us, because we want to believe that the government is good, and people are generally good for the, indeed they are. And we can't even imagine that there would be such evil on the face of this planet. And that's literally what makes us susceptible to being manipulated, to being destroyed. Is we in our hearts, raised by loving parents who instilled ethics and morals and values in us, we can't gather that. We can't conceive of that. And that's why the seats aren't filled. But I want you to know I'm coming back. It's already scheduled. Yeah. Okay. I'm coming back. Mark down your calendars, November 26th. And so your assignment after tonight, should you desire to accept the challenge, is to bring at least 4 or 5 friends so that next time we come, we will have over 1,000 people here. Okay? So at any rate, how I got started in this was the case of the Florida dentist who infected his patients with the AIDS virus. You remember that case? I was the chief investigator of the murder theory of that case after studying it for 3 years. How I got to that position was because there was a company by the name of Henry Schein. It was the largest dental medical catalog supply company in the world, and I was the fellow that the CEO turned to when people were afraid to go see their dentist in the wake of that horrifying tragedy. I was the one that was called upon to develop educational literature to help allay people's fear of AIDS in the dental offices and medical offices, because I was a dentist for 16 years. I had a Harvard master's degree in public health and media, health education, health promotion, and persuasion technologies, and which I used to develop the Overcoming Your Fear of the Dentist audio-taped interactive behavioral therapy program and a host of other self-care packages. I was into consumer awareness, you see. I still am. I believe that empowered with information and choices, we can create a healthy, happy, and successful destiny for ourselves. So that's my love. So that I started investigating that case. What did I find after 3 years was that the government had deleted, covered up the most incriminating evidence against the dentist. They had committed scientific fraud. They did not tell us that he was angry at the government, for he believed that they had created the virus from which he was dying. They did not tell us that he believed in what was called the World Health Organization theory of AIDS. The belief that the World Health Organization and their contaminated vaccines had something to do with the spread of HIV to gay Americans and black Africans. They did not tell us that. I just stumbled on the fact when I did a personality profile on the dentist. I just stumbled on the fact, and then after I went to the FBI and got a hold of their behavioral science literature and how they evaluate and investigate these types of cases, I just stumbled upon the fact that they did not tell us that he maintained a personality profile consistent with those of 36 serial killers studied by the FBI. So consistent that you'd have to be an idiot with anything less than a grade 101 level of behavioral science, psychological criminality, criminal psychology training, to not get it. The guy was an obvious organized serial killer. And based on the FBI's methods and materials, what I did, and this is the #1 investigation question, I called it the Deadly Innocence Investigation, because that became the title of my 9th book, which is called Deadly Innocence. It was based on 3 scientific reports in this case. The last one was in the British Dental Journal. It was called Murder and Cover-Up Could Explain the Florida Dental AIDS Mystery. It's a peer-reviewed scientific journal. And I had to conclude, using the FBI's official methods and materials, that this dentist, based on all the evidence, was inclined to inoculate his patients with HIV-tainted anesthetics in order to express his vendetta against the government. for creating the vaccine and the virus from which he was dying. But you see, that was the only thing that made sense. In essence, he created a crime, a mystery, I had to conclude, that could not be solved without implicating the government. You know how, like the Unabomber, these are smart people. Dr. David Acker, the Florida dentist, was scientifically trained. Military dentist for much of his career. And like the Unabomber, you know, he publishes his manifesto in the New York Times and Washington Post and says, here's what I believe, you try to get me. They manipulate the authorities. They give them catch-22s. They play games. Those are the games that these people play. And Dr. David Acker played the game. Said, I'll create a crime, a mystery that cannot be solved without telling the truth, what I believe the truth to be about HIV/AIDS. So that's why the government covered up the evidence. That's why they told you that it wouldn't, he was angry. You know, they made it sound like he was angry at homophobic society. They blamed it on all the homophobes, you see. And they said he was angry because it wouldn't be until, and they said, teenagers and grandmothers get this disease that the government would do something like create a cure. See, that's all true. But consistent with counterintelligence propaganda standard technique, you see, they present 80% truth, peppered with 20% lies, or they leave 10 to 20% of the most important facts out, that if you knew what those facts were, it would present a completely new picture of the entire story. In essence, the entire story, the truth, the vendetta, the principal motive was screaming loud and clear in the legal testimony. He believed that he was dying from a virus that the government had created. I had to do some research in it. I did not know what was the World Health Organization theory of AIDS. The dentist had gotten a hold of a videotape called the Stryker memorandum. How many of you have heard of the Stryker memorandum? Okay, a 2-hour videotape, horrifying videotape, 1986, produced by Robert Stryker, MD, PhD, who studied the origin of AIDS with his brother, an attorney, and believed that it was a recombinant of the bovine and sheep Visna viruses. And then Stryker said this was spread by World Health Organization vaccines to Africa. And David Acker got that material. Hours in heated debate. He argued with his best friend, Edward Parsons. So that's basically the story of deadly innocence. But at the end of that investigation, although I felt that I had solved, and I think anybody who reads the book Deadly Innocence can agree, solved the mystery, what the authorities claim to be one of the greatest mysteries in American medicine of all time. They call it, quote, an unsolvable mystery. Not unsolvable, it's very clear. So but at the end of that, I have one document that I could not reconcile, and it troubled me, as it would trouble you. As it sat on my desk for 8 months, and I lived in denial about it. It was in Dr. Acker's possession. Stryker had sent it to him. It was a 1970 Department of Defense appropriations request for $10 million. $10 million. For a 5-year study to develop immune system ravaging microorganisms for germ warfare. And in fact, this document stated, when you read it, that the National Academy of Sciences, the National Research Council, the most esteemed of all scientific agencies in America, not only informed the Department of Defense in 1969 that it would be possible to produce, quote, a new infective microorganism that could differ in certain important aspects from any known disease-causing microorganism. And most important of these is that it would be refractory to the immunologic and therapeutic processes upon which we depend to maintain our relative freedom from infectious diseases, end quote. In other words, what they are looking for was a, quote, super germ. That word, end quote, was in the congressional record that could wipe out the host, the human being's defense system, the immune system, which defends us against infectious diseases, leaving us susceptible to the opportunistic infections like tuberculosis, like pneumonia, like candidiasis, and cancers. And so the National Academy of Sciences, National Research Council not only formed DOD, that for $2 million a year over 5 years they could do it, but that they would help them to do it. And who was it on that panel? Primary individual on the National Academy of Sciences National Research Council at that time, the world's leading biological weapons experimenter expert, Dr. David Baltimore, who just 3 weeks ago became Caltech's president, sent from MIT to become Caltech's president. And what did he say? I was in Pasadena a couple weeks ago. I warned people from Pasadena. I said, you guys should go out and picket. Picket Caltech. Because what he's saying came right out in the Herald Tribune of Pasadena, saying that he now plans to make the Pasadena, LA area a corridor for biotechnology research. Wonderful. I'd like to have that in your neighborhood. So, at any rate, it was with that document sitting on my desk for 8 months, troubling me. Ultimately, I moved from denial to curiosity. And I said, okay, I'm going for it. I go back into Harvard's Callaway Medical Library, where I did most of my postdoctoral research, and I, just because it was called the World Health Organization theory, go to the old stacks. You see, 1965 to 1975 became my study period because the money kicked in around 1970. So I figured I'd go 5 years before that, 5 years after that. That became my decade study period. And so I go to the World Health Organization Chronicles and start reading what they were doing in 1965. And what they were doing was they had an evolving viral research network. Had been evolving since the late 50s. And I learned some very interesting things I didn't know as a public health professional. Dental public health I had been involved in for over the previous 5 years. 4 of those 5 years I had actually presented scientific papers at the American Dental Public Health Association meetings. And I found it extraordinary that with my background, my involvement in public health, I knew little, if anything, about the World Health Organization. The fact that they were kind of like the godfathers to the pharmaceutical industry, I learned. That they largely set the standards, or they set the standards for all the pharmaceuticals, including vaccines worldwide. And that they largely impact which drugs and help determine which drugs remain legal, which ones remain illegal. Found that interesting. I found it interesting that they had a viral research network developing since the late 50s. And they published that by 1970, they had, quote, isolated more than 70,000 strains of different viruses. And it took me a couple months before I realized that when they said isolated, what they really meant to say was recombined, mutated, hybridized. And that they published distributing these more than 70,000 different strains to well over 500 research laboratories throughout the world. And their principal distributors included none other than the National Cancer Institute in Bethesda and the Centers for Disease Control in Atlanta. So it was when I saw the National Cancer Institute in Bethesda that a red flag came up for me. Because this, you see, is the home of the infamous Dr. Robert Gallo. Now, many of you know Dr. Robert Gallo. If you saw the HBO special based on the book The Band Played On, where Alan Alda played Robert Gallo, the fellow who allegedly discovered the AIDS virus in 1984, then got involved in the French-American AIDS fracas with Luc Montagnier. You remember the French guy from the Pasteur Institute? And then it was determined that Gallo actually committed scientific fraud. He cloned Montagnier's virus, called it his, and then America patented everything having to do with AIDS research and AIDS testing. And the French got mad. And they started to sue the United States, and it lasted about a decade. And a couple years ago, they settled the suit, and around the same time, President Clinton pardoned Robert Gallo for scientific misconduct and fraud. Now, whenever does a president of the United States pardon a health scientist? It's a little strange, isn't it? By the way, the pardon was for misconduct on a completely unrelated matter, another matter. In fact, it had been the 4th time in 3 decades that Gallo had been investigated for scientific misconduct and or fraud, and let go all times. So I thought to myself, well, this is the chief retrovirologist, AIDS research, AIDS development perhaps. Let me go back into Harvard's Callaway Medical Library, pull Index Medicus off the shelf, and see what I find. And what I found was about 40 articles that Gallo published with his colleagues between 1967 and 1974. Nothing in 65, nothing in 66, 1967. There were 2 publications by Gallo and his colleagues. 68, there were 4. 69, there were 2. 70, there were 3. And 71, oh, just coincidentally, you see, that when the Department of Defense money kicked in, Gallo's laboratory output more than doubled. Oh, it's just a coincidence. Maybe. So I started to read these articles, locked myself in a room for 3 weeks. It's kind of difficult to get through, you know, virology jargon. It's a whole language in itself. So you have to, you know, cross-reference textbooks and try to get the terms defined. But I did that. I came out pale. And I found that approximately a third of what Gallo published, he co-authored with an organization called Lytton Bionetics, or Bionetics Research Laboratories. For example, here you see, this is from Nature, the journal Nature, December 5th, 1970, where you see the name Dr. Robert C. Gallo from the National Cancer Institute, and you see Robert C. Ting, T-I-N-G, from Bionetics Research Labs, Bethesda, Maryland. And what are these folks studying here? Well, it's a very unique enzyme. This is the technical name for what's called reverse transcriptase. RNA-dependent DNA polymerase. Now that's difficult for most of you, so let's just call it reverse transcriptase. And what are they studying this thing? They're studying it in human, acute, that's quick-acting leukemia cells. Now, what is reverse transcriptase? It is an extraordinarily unique enzyme that they're studying that literally calls for life to operate in reverse. You see, traditionally, God created all cells on this planet, all life, to operate on the same miraculous system based on a blueprint of life called DNA. And it is the DNA that presents the code for the RNA production. DNA goes to RNA, and the ribonucleic acid then functions to bring amino acids to the protein manufacturing station of the cell called the ribosome. And it is here where the proteins are assembled into new cell structures, and then as a result, healthy cells develop. So the process that God planned was DNA to RNA to proteins to healthy cell parts to healthy cells. That's how it's supposed to work. Now this particular enzyme and type of virus, the AIDS virus, this reverse transcriptase is the enzyme that makes the AIDS virus tick. It's the enzyme that makes the leukemia virus tick. It says, no, 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 we're not going to have this anymore. We're going to go backwards. We're going to take a single strand of RNA that the virus presents. And then the virus, with these little ball-like structures called GP120-like proteins, they attach like lock and key fashion to the T cells or the CD4 helper cells, and then injects this single strand into the virus, and then because of the presence of this unique enzyme called reverse transcriptase, it causes this RNA to get translated into DNA, so that you now have a new DNA strand, and because of the presence of this little scissors-like enzyme called an endonuclease, it functions to cut open the normal, healthy human gene and allows this new DNA, this pro-virus DNA, to get inserted into God's perfect blueprint, so that now the cell gets fooled. Because as it begins to read the code calls for the production of more viruses instead of healthy cell parts. And these now viruses butt off the cell, the new viruses butt off, they seek out additional CD4 helper cells, and that's how the disease process continues. Now, This is what these people are studying in 1970. This is 14 years before this man allegedly discovered the first AIDS virus. This is 9 years before the same man, Dr. Robert Gallo, allegedly discovered the first leukemia virus, HTLV1. And it was Dr. Robert Gallo that said that it was an African green monkey that bit a man, or was eaten by a man, and somehow Portuguese seamen transferred this virus to Japan some 500 years ago. And of course, when I presented this research, I had the great fortune of being the first investigator in the history of the esteemed International AIDS Conference, not far from here in Vancouver, on July 10th, 1996. And presented this research being the first investigator to defend a scientific paper saying that the AIDS virus and its relatives from the simian immunodeficiency viruses from the monkeys are undoubtedly laboratory creations. Some of my esteemed colleagues, epidemiologists and virologists, said, "So what, Dr. Horowitz? So they were studying this enzyme at that time. That doesn't say anything." I said, "Well, okay." I said, "Well, how about the fact that Gallo's co-authors are from Bionetics Research Labs, which just happened to be 6th on the list of major Army biological weapons contractors for that year?" And they said, "Oh, that is interesting, that is interesting." Also notice New York University Medical Center. This is from the United States Congressional Record. You see, this is New York University Medical Center. Remember that. I'm going to come back to that. There'll be a quiz at the end of this program. And those who don't get a passing grade, your lives might be in jeopardy. What else did they report? They reported taking, these were the preeminent monkey virus, primate cancer virus researchers in the world. So they reported their studies for cancer research. And what did they report? Well, they were taking, the experts in taking monkey viruses, like simian virus 40, the 40th simian monkey virus ever discovered, which, by the way, they knew in the late 50s, early 60s that it would cause cancer in virtually every animal it was injected into. And in the 80s, they started to realize that it would cause cancer in humans. And you know what? They suspected in the early 60s it would cause cancer in humans. But you know what? To this very day, the FDA, the Food and Drug Administration, our medical mafia, the United States, turns a blind eye to as many as 100 simian monkey virus contaminants per dose of the oral polio vaccine that you and I are allegedly by law required to give our children. And the reason why they don't clean them up, it seems to be a mystery, but the reason why they don't tell even health scientists the truth, so that physicians never find out, and the public stays in a complete fog, the reason for all this is because apparently their hands are tied by proprietary laws and non-disclosure agreements placed upon them by the pharmaceutical industry. In other words, they are muzzled by the drug makers. So they took this virus and a whole assortment of other viruses, monkey viruses, and human lymphoma viruses that would cause cancer. They knew it would cause cancer of the human lymph nodes, and they started to recombine these with virtually every animal cancer virus known. For instance, their favorite ones to utilize were feline leukemia virus RNA that caused cat leukemia. A whole laundry list of symptoms, virtually identical to what AIDS patients suffer from. Their second favorite was this thing called chicken leukemia sarcoma virus, RNA. And they added that to the viruses, to the mix. And then this caused wasting, immunosuppression, and death. And then when they got to this point, when they had this mutant hybrid here, it still had some difficulty infecting humans. So to get it to jump species now, they cultured it, they reported culturing it in human white blood cells in some studies, and human fetal tissue cells in culture in other studies, so that it would adapt. it would develop those little unique ball-like proteins on the outside membrane, those GP120-like proteins that allowed it to attach to the human cells, be carried across the cell membranes, enter the gene of the cell, and then do virtually everything that the AIDS virus does. So, when I presented this in Vancouver, many of the colleagues, esteemed colleagues, said, "Oh, that seems strange. That seems very strange. How do we know you're not making this up?" I said, "Well, if you sincerely are concerned, then what you should do is look up the references because it's meticulously documented. But to save you the trouble, why don't you take a look at this document?" And then I showed him a document that I had gotten literally from Lytton Bionetics. It was published by Lytton Bionetics. This is a diagram that I've created to explain exactly what they were published in this document. And you see that these were not just biological weapons contractors for the Department of Defense, they were also cancer researchers. They were studying in cancer research 3 different things. The first thing was whether indeed cancer viruses would cause cancer in humans. They didn't know that for sure. They really did not know that for certain, allegedly, until Dr. Robert Gallo in 1978 allegedly discovered the first leukemia virus, HTLV1. So they were trying to experiment on different models. And their principal model, that means they were creating viruses, trying them in human tissues and various animal tissues, and the principal model, and this is one of the many smoking guns in the book, Emerging Viruses, AIDS, and Ebola. You see, because the principal model that they spent most of their time and money studying was none other than the immune suppression, leukemia, lymphoma, sarcoma, cancer complex that never existed on planet Earth in human beings before 1978 with the first cases of AIDS. And that's precisely what they were studying most of their time and money studying between 1962 and 1976 during a largely funded and mostly secret, quote, special virus cancer program, end quote. So here is that to show you what they were doing. Besides looking at that model, they were looking for vaccines to cure viral-induced cancers, and they were looking for early diagnostic screening methods, you know, like pap smears and breast exams, which is good prevention, you know, kind of like you look at these things early to try to diagnose things early, and you can't blame these people for doing that. Here they're trying to diagnose, in this particular study that they wrote about, cancer viruses early. Now, I asked a question to my esteemed colleagues after they read this, and after I explain it, I'm going to ask you in this audience the same question that I asked for some of the world's leading AIDS researchers and virologists and epidemiologists. So here's what they're looking for as an alternative approach now to the detection of possible human leukemia viruses. To begin this research, they take a mouse sarcoma virus and recombine it with a mouse leukemia virus to create a mouse leukemia sarcoma virus hybrid, which they then inoculate into healthy cats to get it to jump species from mouse to cat. By the way, for those of you who are cat lovers, you might be horrified to learn that the earliest researchers said undoubtedly the feline leukemia virus is man-made. Cat leukemia is a man-made condition. Got out in cats, didn't it? So they then had this mouse leukemia sarcoma virus that was infectious for cats, which they then inoculated into a cat suffering from leukemia. And you know what? Success! They were proud to publish that this was successful. They were able to, using this method, diagnose feline leukemia viruses in the tissues early. Next step, they report, culture this same mutant recombinant from a mouse, cat, into human embryonic cells in culture, and then inoculate it into a human being suffering from leukemia. So my question to my esteemed colleagues, as it is to you now, how many of you think that this might be risky to humanity? Raise your hands. Yeah. So the vast majority of you raise your hands, and I want you to know, as a lay, mostly lay audience, you concur with the world's leading virologists and epidemiologists and AIDS researchers, who most of them shook their head as they read this piece. Many of them said, I can't believe they did this. Several of them said, this is extremely risky to humanity. So I presented this, I presented my thesis. This is basically what my thesis looked like. And because we had sent out press releases to the news media, you would probably be interested to learn that most of the Canadian press at least covered the press releases and invitations that we sent out. In fact, I made it onto every wire service in Canada, every news radio program in Canada had a little blurb about my presentation. The province had a very nice feature article, although they didn't show up yesterday. Nothing in the Sun, but we did manage to have CBC come and interview me, but they never showed the interview. But that's far, far better than the American counterparts. You see, Associated Press, UPI, AP, and Reuters were all invited with the same invitations, and they didn't show. They didn't even come at all. Americans learned nothing about my presentation. And frequently, sometimes, if it's incriminating to the Canadian government, you Canadians hear the same silence. Right. So for those of you who, at this point in time, don't believe that the media is heavily rigged, you better think again. It's time to think again. But at any rate, while I was in town in Vancouver, there was a documentary film crew there watching me, taping me on Fanny Kiefer, who is one of Vancouver's leading talk show hosts, and they were filming a vaccine documentary. And at the end of the documentary, at the end of the filming and Fanny's show, the producer said, Dr. Horowitz, did you know that Dr. Robert Gallo was in town this week? And I said, no, I didn't know that. And she says, yes, indeed, he's giving a public presentation at the Robson Square Conference Center, and I believe that the public is invited to ask questions. Would you like to come and join us and ask Dr. Gallo a question? So, of course, those little horns pierced my scalp. And I said, sure, I'm game. So the next night, in front of about 300 Canadians, the Canadian press, and a documentary film crew, I asked Dr. Gallo the following question. I said, Dr. Gallo, are you at all concerned that your early research in taking monkey viruses like SV40 and recombining them with animal cancer viruses like feline leukemia virus RNA and chicken leukemia sarcoma virus RNA might have given rise to the AIDS virus or AIDS virus relatives that might have contaminated chimpanzees shipped by your colleagues from Lytton Bionetics to the vaccine manufacturers at Merck Sharp and Dome. He got real angry. Woo! Woo! See, man. Steam poured out his ears. His eyes turned like fire. He started twitching and shuddering. Oh. First he denied it. He said, no, we don't know what you're talking about. We never did anything like this. No, you don't know what you're talking about. I said, excuse me, Dr. Gallo, because I had a microphone. There's power in a microphone. I said, excuse me, Dr. Gallo, I could cite your papers. He says, well, if you could cite my papers and you've got a paper I don't know about, you go right ahead. I said, okay. I said, Adamson, Herrera, Gallo et al., Proceedings of the National Academy of Sciences, 1970. Dr. Gallo, you might recall even that you published presenting this research in front of NATO military scientists in Mol, Belgium. That's enough. That's enough. Indeed, yes, I was in Mol, Belgium. I was in Europe at the time. He said, I can remember it because it was the first time I was in Europe, and I want you to know that NATO meetings, however, fund all sorts of meetings. They fund meetings about motherhood and fatherhood and sisterhood and brotherhood. And you really don't know what you're talking about. You can't mix these viruses. In fact, what you're talking about is like mixing kiwi and tutti frutti with grapes and apples and oranges. And with that, many in the Canadian audience laughed. And then he said, and you should go back and get a grade 101 level of biological science training, high school level. And with that, many in the audience applauded. Wow. And I sat down, even though I threw in the fact that he was manipulating the leukemia, lymphoma, sarcoma complex models. He didn't want to hear about that. But anyway, so he then hears from somebody else who says to him something about Dr. Peter Duesberg's work, and Gallo, in a huff, silences him, and then he kind of like realizes that he's lost it completely, and he apologizes to the audience saying he has a low-grade fever, he hasn't been feeling well today, which is a standard public relations ploy that people lose when they lose their cool. They're trained to do that, you see. And then a couple minutes later he left in somewhat of a huff, surrounded by a couple very, very elegant-looking tall gentlemen, one of which passed out a business card to a colleague, and it said Vice President Helen Knowlton, public relations firm. Who, if you know anything about public relations, you know that Helen Knowlton is one of the most esteemed and busy public relations firms in the world. In fact, Helen Knowlton was the public relations firm that the George Bush administration called upon to persuade Americans who, like Canadians, do not like sending our men and women off to die in foreign lands. And we had this thing called the Gulf War coming up on us, and you see, Americans needed to be persuaded that it was important and that we were really fighting a bad, evil foe. So Hill and Knowlton dreamed up this fabulous public relations ploy. They got a hold of a Kuwaiti official's relative, a young woman, who alleged that Saddam Hussein's elite guardsmen were torturing and maiming babies and pulling them out of their incubation chambers and throwing them out from windows. And the American people, who are loving people, were horrified and disgusted at this. And then George Bush announced, this aggression will not stand. And Americans said, rah, rah. And then they inoculated 700,000 of our finest men and women with vaccines. Only to find out, when they came back, 200 to 300,000 of them now suffer from Gulf War syndrome, 20 to 30,000 of them were dead. And what Hill and Knowlton did was a complete scam. It only came out afterwards that it was a complete hoax. There was a psychological warfare boost. And what they don't tell you now, first you have the Pentagon denying that Gulf War syndrome even exists. Then when they couldn't deny it any longer, they claimed it was all in their heads. Then when that wouldn't work anymore, the Pentagon hired some puppets to claim that it was drug interactions only. Drug interactions. They were using experimental drugs, new drugs that recombined and caused chemical sensitivities in these people. And you know, the data supported a little bit of that, but it really didn't explain all of that. And then you had the CIA finally saying, oh no, what about these people like Joyce Riley and Garth Nicholson and others who showed that the Pentagon was covering up the fact that the troops were exposed to raining biological and chemical weapons as the Scud missiles were exploded out of the sky by the Patriot missiles. The troops were exposed there, and then the weapons dump explosions when the wind pushed the chemicals and biologicals over our troops. So the Pentagon had that information, but oh, they conveniently lost those records as they exploded in Oklahoma City.
Ah.
And so the CIA says the Pentagon is covering up, and the Pentagon says, no, the CIA doesn't know what they're talking about. And you see, this is how counterintelligence propaganda, smoke and mirrors, is waged. Neither one tells you that the most plausible initiating factor of Gulf War syndrome, like AIDS, is contaminated vaccines. But you see what else they're not telling you? Remember I said 80% truth? The formula is 80% truth, peppered with 20% lies, or 10 to 20% that they leave out that would give it a completely new picture. Here's the 10 to 20% that they not tell you. You see, vaccines are the only plausible explanation of why it would be that some men and women who went over to the Gulf, got exposed, did not develop the symptoms, versus others who did not even go over to the Gulf War arena, got Gulf War syndrome. Why? Because when you produce a contaminated vaccine lot, some lots are more contaminated than others. And when you get a single dose from a contaminated vaccine lot, some doses are more contaminated than others. And when you happen to get a contaminated dose, then what happens is your immune system goes bluey. It blows out your immune system. So that now what happens is that you get exposed to drugs with a reduced immunocapacity, reduced defense, and you develop multiple chemical sensitivities. Similarly, biologicals wreak havoc on your system if it's, immune system dysfunction. So that's what explains it. What they don't tell you also is the fact that the Bush administration was supplying Saddam Hussein's guardsmen in military production labs with not only military weapons, but biological chemical weapons reagents up until 2 weeks before they invaded Kuwait. In other words, they knew that when our servicemen and women went over there, they were going to be exposed to these things. Moreover, what they don't tell you is the fact that much of those vaccines were developed at Panox Biomedical Systems Corporation in Houston, Texas, co-owned by James Baker III, Secretary of State under George Bush, heavily invested in by George Bush, and that Panox Biomedical Systems tested that vaccine on Huntsville prisoners in Huntsville, Texas, and the prisoners developed Gulf War syndrome long before the Gulf War. And then they spread it to the guards, who then brought it home to their wives and children, who then began to spread it to even some health professionals. You see, it's an infectious disease that today is being covered up. So this is who Robert Gallo is escorted out of Robson Square Center. Nice, huh? Would you like to have friends like that? So, shortly thereafter, somebody hands Gallo a flyer on the book Emerging Viruses, AIDS, and Ebola. And he sees on the front of the book cover the name W. John Martin, MD, PhD, and he recognizes it. Not only as among the world's leading vaccine contamination analysts who just happen to work for the FDA, Food and Drug Administration's Bureau of Biologics Director between 1976 and 1980, in charge of testing human vaccines for contamination. But he recognizes Martin also as the guy who rented a room in his house for a long time. And he recognizes the name because he had just been in Martin's lab about 3 weeks earlier. Because Gallo is known for stealing things, you see, like Montaigne's virus, calling it his. He was interested in what Martin and his colleague Zaki Salahuddin, who used to work for Gallo, was up to. So Gallo then flips the flyer over, as I'm saying on the back of the book cover. He sees Garth Nicholson, the hero to Gulf War syndrome veterans, the only scientist out there who's really telling it all as much as he's finding it. As a result, he was politically pressured to lead MD Anderson Cancer Research Center, wherein he was Gallo's counterpart. Gallo knew him well. They both worked under the Special Virus Cancer Program together. And Nicholson writes, one cannot fail to grasp the explosive significance of Dr. Horowitz's work, that biological weapons were developed and field tested, and that now they may not be contained. Cogent, readable, carefully documented work. So Gallo gets home. He calls Nicholson on. Now I hear this from Nicholson. He says, what the heck are you giving Horowitz, this loon, a good testimonial for his book? Nicholson says, Bob, hold it. Wait a minute. First of all, he's no loon. He's a Harvard grad. Second of all, have you read his book? He says, no. He says, Bob, you better read his book. Here's his number. So here's my telephone number. 3 minutes later, my phone rings. Pick up the phone. This is Gallo. My first thought is, I wonder what the world's leading AIDS researcher is doing, calling somebody that needs a grade 101 level of biological science at high school level. So the first thing he says, I want to apologize to you. I thought that was nice.
Yeah.
I want to apologize to you for what happened in Vancouver. I thought that was real nice. He says, I want you to know I'm a humanitarian like yourself. Oh yeah, I'll believe that. He says, I want you to know that I want to help you. I might have some information that you can use to really find out the true origin of AIDS. And I'm willing to help you. I said, that's nice. And then he starts telling me why he's so interested in helping me as such a humanitarian. This goes back to his family history with his sister dying of leukemia. Everybody who knows anything about Gallo has read about this time and time again, and we're tired of it. But I know the conversation's going on. And you know what? I have a tape recorder next to my phone. Ready to go. So I asked Dr. Gallo, Dr. Gallo, do you mind if I record this conversation? He says, for what purpose? I say, well, for publication purposes, of course. I now find myself in the role of an investigative reporter. So he thinks about it for a couple seconds and he says, okay. So I start the tape. I get about 30 minutes on the phone in which he lays out his 4 major objections to my thesis. And I go back to Harvard County Library over the next couple of weeks, and I research his objections, and I want you to know I found them to be absolutely scientifically bogus. Scientifically indefensible. In a scientific group, his information, compared to the documented facts and evidence, he would be laughed out. But I pressed them, because I know the thesis is sound, and when you press Gallo, there's this thing about Gallo. He's known worldwide. He even admits it himself. He's got the greatest ego in medical science. He's always got to be one up on everybody. He knows it's his greatest, weakest point. So I press him. I said, but Dr. Gallo, don't you think it's plausible, but not necessarily in your lab? He says, okay, I do not argue with you. I will not argue with you. It's plausible. But I could tell you, even more plausibly, how something like this might have happened by accident at that time. Now, isn't that nice? More news that you're not reading. World's leading AIDS researcher says that the epidemic might have been an accident. How nice. So anyway, I sent him a copy of my book. I said, Dr. Gallo, if you can make any additions or corrections to improve the quality or content of this work, I would be grateful if you would send it to me. And also, if you can tell me more plausibly how something like this might have happened by accident at that time, then I would certainly appreciate that information, as I'm sure maybe 30 million HIV/AIDS patients worldwide might also. I didn't hear back from Gallo. So a couple of weeks ago, I called Gallo, and I'll tell you in a few minutes what he told me. I want you to know that virtually everything that you know about AIDS is a deception. In fact, I want you to know that virtually everything that you know about everything is a deception. And to prove my point, I'll just take one simple example of the notion that the AIDS virus doesn't discriminate. You've heard that, haven't you? And probably most of you with active intuitions kind of sense, you know, I'll bet it discriminates. In fact, I'm going to show you right now that the AIDS virus discriminates in virtually every high-risk group even. It discriminates. You were told all sorts of nonsense like white women. A couple years ago, the Centers for Disease Control released a new report saying that white women were the fastest growing risk group for HIV infection. Remember that? ages 23 to 35. Again, to push the concept that the AIDS virus doesn't discriminate. I want you to know that that was a total twist of the data. It was a totally misleading communication based on the little data that they had, that they twisted for their political agenda. And when they twist scientific data, it's no longer scientific, it's garbage. And that's what they put out. So here's one of thousands of studies, literally, that you can evaluate and you can begin to think. This is now lifetime needle sharing experience when HIV antibody prevalence among IV drug users in 6 US regions. And the blue represents needle sharing, which you are told is the risk, right? That's the big risk, you're told. You're told that, you know, you share those bloody needles and you get a little blood with HIV in your system. Baby, you better start praying, right? So the red is the seropositive, HIV positive number, percentage. Now here you see in San Antonio, for example, 99% of the IV drug users share their bloody needles. In other words, these are the dumbest IV drug users in North America. But despite that massive ignorance, a mere 2% carry HIV. Now that's interesting. In San Francisco, you look at 97% similarly ignorant IV drug users. It jumps up a little bit to 10%. But here in Baltimore, an extraordinary thing happens. It jumps way up to 39%. You know, common sense might suggest to epidemiologists that something strange might have been going on in Baltimore, Maryland, around 1974, 75, 76 maybe. But you know what